Again, I am Dr. Holly Lucio. I am a naturopathic doctor and senior medical advisor here at FullScript, and I am so happy you've joined us today. The focus of this particular webinar is on mitochondrial health. And our first session, we're going to be focusing on how mitochondria are damaged and hallmark ways to mitigate this damage using metabolic and micronutrient testing. All right, next slide. So leading today's first session is my good friend, Dr. Michael Chapman. He is a naturopathic doctor and a graduate of Bastyr University in Seattle, and he's the director of product innovation at Genomic Diagnostics. And just by the way, I've been using and familiar with Genova Diagnostics ever since they were Great Smokies back in the day. So I'm super excited about this. Dr. Chapman oversees the research and development of new test profiles. So the testing has evolved, the quality has stayed the same, and plays a key role in educating and consulting with healthcare providers. So before we get started, obviously, I'm going to repeat these if you've been with us before. Housekeeping notes: submit your questions in the chat and we'll address as many of them as possible. Usually we have so many questions, we can't get to them all. The webinar will be recorded and a copy of the presentation and the slides are going to be available for download in the files section of the webinar platform. And again, thank you so much, Dr. Chapman, for joining us, and take it away.
Awesome. Thank you so much for having me. One of my favorite things to talk about, this mitochondrial dysfunction, mitochondrial health. Anytime I get into some of these little cellular processes, I get super excited. So I'm going to do my best to get through as much information and translate it, make sure it makes sense, because we do get into the weeds a little bit when we're talking about things like mitochondria and whatnot. But without further ado, starting high level, I like to just reframe, and a lot of us who are here watching this understand that there's lots of different ways that we can think about disease, and probably very familiar with this conventional model, this siloed model where we're talking mostly about different organ systems and how they function individually, and less talking about how those organ systems really kind of interconnect and interrelate. And beyond that, oftentimes with conventional approaches to medicine, we're talking about this diagnosis and treatment model. And it's really tricky to look at health this way because, oftentimes we start with the diagnosis and we can use a test to diagnose a condition, or oftentimes it's more of a umbrella list of symptoms like an IBS, right? It's just a sort of conglomeration of these different systems. And then we end up with some of these non-specific labels, like you can see here, R14.1 gas pain, right? And none of these are actually telling us anything about why. And unfortunately, even though we think of this as being kind of the conventional model, even more of our precision-based, our integral, our functional approaches, our natural approaches, oftentimes reflect that similar sort of model too. And so when we compare that, or I would say contrast it to another way to look at it, I like to think about one example of, well, what's going on in the cell, right? We have this cell, all of our tissues are made up of cells, all of our organ systems are made up of tissues. So if we really zoom in and talk about the health of the cell, we're going to be understanding so much more about the health of the system. And in particular, today, we're going to be talking about mitochondria specifically, which are these power plants. If you think of the cell as a city, then the mitochondria is what's producing all the electricity for everything that the cell needs to accomplish. And you don't want to be running out of energy either in a city and having blackout spots or within a cell, because you can imagine all of these other functions of the cell are going to take a hit, and we don't want that to happen. And so if we move towards this model of cellular health, then we can really focus on one of the major things is the mitochondria. And what does it mean to even have mitochondrial dysfunction? How does a mitochondria get damaged, right? And so starting high level, some of the initial factors that create mitochondrial damage, mitochondrial dysfunction are primarily, number one, oxidative stress. And by oxidative stress, if you're less familiar with that term, we're talking about damage. And oftentimes this is free radical damage. We have all of these toxins and things that can run around, run amok. That can be pesticides, that can be metals, that can even be the own reactive oxygen species that mitochondria make. So remember that mitochondria, in the process of producing all this electricity, is kind of a dirty job. So even in doing that, they're actually creating a ton of oxidative stress that they then have to mitigate. And if you are pushing your mitochondria, say through tremendous exercise, exertion, or even infection, you're more at risk of creating more free radicals that the mitochondria has to then handle. And it handles this by using nutrition, right? And so if you have a nutritional deficiency, this is another factor, another trigger for the mitochondria starting to break down. What's more than that? Inflammation. So pro-inflammatory cytokines can alter mitochondrial function as well and leads to additional damage, additional dysfunction. And sure, there's genetic mutations that are also going to put people at higher risk for mitochondrial dysfunction, but those top three bullets are the ones that we really have a great opportunity to correct and take care of. And so that's what, we'll be talking about in a little bit here. But on a higher level, on a bigger, we're hearing a lot about longevity, right? And aging and what it means to be aging. And if you look here, there's several papers that have talked about this oxy-inflammaging theory. And really, like I just said, it's oxidative stress, it's inflammation, and to a certain extent, metabolic dysfunction. These are the main things that are driving aging. And these are the main things that also drive mitochondrial dysfunction. So you can see how interlinked the idea of the mitochondria are with longevity. You want to have good longevity, you want to then have healthy mitochondria. And then it's just another paper, demonstrating, discussing this oxy-inflammaging hypothesis, and that those are the major drivers of overall aging theory. So the next step, just getting a little bit into the weeds around mitochondrial dysfunction, is as the mitochondria start to be damaged and are attacked by these free radicals, what actually happens is the complexes in the mitochondria that are responsible for creating energy in this electron transport chain, they start to become weak. And ultimately, there's what's called this development of pores. The mitochondria signals, hey, I'm sick, I'm having a hard time. And then ultimately, that can lead to the cell itself taking out that mitochondria, or even the cell death because it runs out of electricity. And so you can see how, if that's happening throughout the body, that is a major, major bad sign. And so ultimately, problems with the cell are going to be decreased cell energy affects most of the most active tissues in the body. So we're talking about your brain, your heart, your kidneys, your muscles. And so you're going to have symptoms like fatigue and cognitive impairment, muscle weakness. Moreover, damaged mitochondrial proteins actually trigger an inflammatory response. So if this mitochondria breaks down so much that it's releasing some of its proteins, your immune system will identify that and create more inflammation in the body. This all swirls around, and this is the fuel that fires biological age. So what if you could test for all that? There's lots of different ways. I'm not going to spend a ton of time talking about all the different tests for mitochondrial function, but most of them are what we've seen are telomeres, which is not necessarily a direct measurement of mitochondrial function. There are some things that can be done more in a research lab that require like muscle biopsies or blood cultures. Not really great for everyday use with our patients. But if you look at something like a test that measures your entire system, then part of that is going to be talking about some of the things we just discussed: inflammation, oxidative stress, mitochondrial function, ATP production. These are all going to give you signals and signs, and then guideposts of how to actually handle it and mitigate it. Where are the actual true problems? And so what I'm showing here is Genova's NutriVal. It's also very similar to the metabolomics test. And this is a cellular health test at its core. And you can see right on the front page, we have a scoring system for oxidative stress and how that impacts mitochondrial function, as well as inflammation, toxic exposure, right? We talked about those are things that can also trigger damage to mitochondria. And then also methylation, which I'll mention here in a little bit, is actually related to the concept of mitochondrial function as well. So again, oxidative stress, we talked about this. Free radical damage certainly has been associated with lots of clinical conditions, including metabolic dysfunction, cardiovascular risk, cancer risk, right? We don't like free radicals. It's not good for the system. But furthermore, omega balance. So omega-3s, omega-6s, this is a good indicator for how well the system's able to tolerate inflammation. Exposure to heavy metals, plastic, petrochemicals, not great. And then methylation imbalance is another thing that we're focusing here on the front page. But what we see is that not only do we have these scores, these identifiers for problems, the next thing is how do you fix it? And these recommendations here are based on all the same things that we saw on the front page. So if you have a lot of oxidative stress, naturally there's going to be a need for additional antioxidant support. If you have needs for methylation, then we're talking more along B vitamins. They're all kind of linked together. Here's the problem, and here's the potential solve for that problem as far as we're seeing. Obviously, the test is not sitting in front of your patient, but it's doing as best a job it can to try and get to some of these underlying problems. Won't spend a whole lot on the nutrient need overview. Just to note, if you happen to do a vitamin D as part of that, then we would also give a recommendation around vitamin D. Amino acids are at the bottom of this page too. And amino acids are critically important as it relates to mitochondrial dysfunction because they're primary building blocks for glutathione, our main body's antioxidant. And so that's why I bring them up a little bit here in this context. We do have really great information, educational resources on the report for patients. Not only are we talking about supplements, but we're also talking about, hey, at the bottom here, there's a lot of foods where you might want to look. If you have a high score on a need for one of these nutrients, also consider adding more of those foods to your plate. Okay, so getting into some of the weeds around the test too, how do we come up with all of these nutrient recommendations? How do we determine mitochondrial function for this? Well, a lot of it comes from organic acids, which organic acids are generally, they're all these little intermediates. It's almost like a person's biochemical fingerprint. But as it relates to mitochondrial dysfunction, we start here with this page around the Krebs cycle and the energy systems. And really what we're doing here, if you take a close look, is we're saying, hey, not only does the mitochondria, all of these sections within energy production take place inside the mitochondria. You have ketogenesis, you have this Krebs cycle, you have the electron transport chain. They're all critical. And if there's issues that we see there, then it's going to demonstrate to the provider, hey, we're seeing some evidence of mitochondrial dysfunction. And the way that this can work too, from a nutrient perspective, is just take an example of this one little pathway, right? You can see here fatty acids, the fats that we eat ultimately go through beta oxidation if we're using them for energy. That requires magnesium and vitamin B2 as cofactors. But if those are not there, then we have these little byproducts that we see shoot off. And so if you see all of this runoff into these byproducts, it's a general indication, hey, maybe we need some more magnesium B2 going on here. And so that's kind of the way this whole page works. And that's just a demonstration of how we're arriving at some of these more cellular analyses. And also, how do we know we have these needs for these additional vitamins, nutrients, minerals? But furthermore, like I said, so much of this is about the electron transport chain, because that's what makes the energy. That's what makes the ATP. And so what you see here is CoQ10 and glutathione are tremendously powerful antioxidants that are constantly trying to reduce and mitigate the oxidative stress that the mitochondria themselves make, right? To make energy, it's kind of a dirty process like a factory or a power plant. And so the glutathione, the CoQ10 help to clean that up. And so if it's not doing a great job, we have two markers on this test that are indicative of now the free radicals have gotten loose, damaging fats, they're damaging DNA, and that's listed here as lipid peroxides and 8-OHDG. I'll mention them here on this upcoming slide as well. So one section of this great report around mitochondrial function is about organic acids. Just honorable mention for some of the other sections, because organic acids is like an entire study in and of itself. There's these main categories. The first main category is around malabsorption and dysbiosis markers. Are we seeing a trend that suggests some type of perhaps dysbiosis or bacterial overgrowth? You can get some reads from organic acids in the urine, even looking at that. But here is the meat, right? This is what we just saw with that Krebs cycle page and the electron transport chain and all of that. These are our cellular energy mitochondrial markers. There's a couple ones on here I really like, so I'm going to highlight them. This alpha hydroxybutyric acid. You may have heard of beta hydroxybutyric acid if you're into ketosis or ketogenic diets. That's a ketone, but this is a little bit of a very similar type of a chemical molecule, I guess you'd call it. But the research has shown that it's a marker for overall metabolic dysfunction and poor lifestyle habits, things like smoking, alcohol, sedentary lifestyle. So not that I want to have a lot of, gotchas on a test. This can be one of those really interesting ones. Another honorable mention here is beta hydroxy beta methyl glutaric acid. That's a long one to say, but it's part of the pathway that takes cholesterol and ultimately it's part of the pathway that creates cholesterol as well as CoQ10. So we look at this marker as an indicator for that great antioxidant CoQ10 as well. And if somebody's on a statin, is there, a problem there? So We also have these additional vitamin markers. These are all directly kind of wired to needs for B vitamins. And it's similar sort of way, right? We know a pathway is strongly dependent on B12. If that B12 isn't there, then we see a runoff into a side pathway, and that gives us some indication of a need, a functional need, right? We're not measuring B12 in this case. How well is that B12 making these pathways almost like highways, right? How well is the traffic flowing? If it's not flowing well, that's an indicator for a need for that nutrient. Same idea here around a couple other ones, folate marker and branched chain amino acid metabolism, all very interesting, not as relevant to mitochondrial function and mitochondrial health, but certainly relevant to things like methylation and other B vitamin dependent issues. We also have a section on neurotransmitter metabolites. Not going to spend a whole lot of time there, but here we go. Toxin and detoxification markers. These in the middle here, alpha keto phenylacetic. This is from styrene plastics. The next one is from gasoline additives and other petrochemicals, right? So we're getting more indication of potential free radicals that could be running around damaging things. And then I mentioned this before when I showed that little picture of the electron transport chain. We measured glutathione and CoQ10 to see if we have the amount of antioxidants we need to support the cell. But then if we don't have that support, then we're measuring oxidative damage, things like lipid peroxides, which is damage to fat, and then 8-OHDG, which is damage to DNA, something we really want to mitigate at all possible costs. So amino acids are the next section on the report for this. Again, it's the NutriBio or the metabolomics. But the amino acids here, it says FMB at the top. You could also run it as a plasma sample. People ask questions about, well, what's the difference? Well, if you're looking at amino acids in the urine, a little bit more reflective of the last couple of days. A plasma example tends to be a little bit more of a steady state of the amino acid status. So when we're thinking of amino acids, really it's the question, is my patient, or am I, if I'm taking the test, getting enough protein, right? Am I meeting my body's needs? Because these amino acids, remember, they're the building blocks of every protein in our body, and including that really powerful antioxidant glutathione. So that's what we're doing here. We're looking at these and saying, hey, is this trending down the middle, these essential, non-essential amino acids, or do we need to up somebody's protein intake? Or if their protein intake is good, is that an indication for digestive support, right? Maybe they're eating plenty of protein, but they're not digesting it. And we actually have a couple markers that can give you some information into that. Got some, I call them meat markers to be simple, but things that come from chicken, turkey, fish, or even your own skeletal muscle tissue if somebody's catabolic. It's sort of a meat turnover marker. But if somebody is eating a lot of meat, and I can see that on this section of the test, but their amino acids are in the toilet, then I have a much greater suspicion that they're not digesting and they need support there. So a lot of great information on this section. We spent a lot of time on the organic acids, but don't forget about the amino acids. And then what about fats, right? Our fatty acids. Don't sleep on the fatty acids either. One thing to note, we always typically look at fatty acids in either red blood cell or whole blood. And we're looking at this as like almost like a big pie chart. Okay. So we have our omega-3s that we've heard so much about. We've got omega-9s, saturated fats, we've got omega-6s, we've got all these categories. And we're saying, well, what is the highest percent? What is the lowest percent? Are these percentages off, right? You've probably heard of like the omega-6 to omega-3 ratio. And that's saying, hey, because oftentimes our current standard American diet is high in omega-6s and low in omega-3s, we're going to be imbalanced there. And if that happens for a long time, we talk about, is that shifting the type of inflammation that's happening in the body? And so moving on, the last couple sections of this report, we're looking at different elements like magnesium, potassium, zinc. These are all important. Zinc is critically important in glutathione production. So is magnesium as well. And then toxics like lead, mercury, arsenic, cadmium. And then one last thing to think about too is that, you know, when we're going back and just recapping everything about mitochondrial dysfunction, I know that was like a ton of information. We've talked about the main things that drive it. There's oxidative stress. And so if you do a test like this, you're getting a great lens into oxidative stress. I mentioned the lipid peroxides, that 8-OHDG, which is DNA damage. We look at antioxidant capacity, right? Do they have glutathione? Do they have CoQ10? Do they have all the nutritional elements that they need to recover and repair? and to produce energy as well. Remember, producing the mitochondria make all this energy. That requires a ton of B vitamins to do that, especially in our athletes, right? They're producing so much energy all the time. They need so much support from a nutritional perspective, a B vitamin perspective. And then lastly, are there things that are making the situation worse? Is there toxicity? Is there inflammation? And I know probably had a ton of clinical claims and things of that nature, and a lot about these biomarkers. Just a reminder, on our website, we have a NutriVal metabolomic support guide, got over 1,000 references in it. These all came, and a lot of them are also in the textbook of natural medicine as well. So these are really great resources to have. Like, I want to see where he said this was a marker for fat damage. Like, if you have any questions, this is a great resource to go to. Everything is completely fully cited. We also have weekly group educational consults if you want to learn more, if you want to hear this information in more detail. And then if you have an account and you have any sort of question whatsoever, you can always call this number and talk to one of us. And shameless plug, wow, so shameless. The Lab Report podcast, this goofy guy here on the cover, and my inimitable co-host, Patty Devers. We do mostly a weekly podcast, try to keep it fun, talk sciencey, talk fun. So, if you're into podcasts, you can check that out too. And with that, thank you so much. Thank you, Fullscript. Thank you all for paying attention to me for this 20 minutes, and any questions, I am happy to address them.
All right. I think I'm coming back. I'm with you. Oh, holy moly. Dr. Chapman, you went through that in great form in the time that we had. What are you doing for the next 4 hours? Because I love this test. But I have to say, one of the questions I do want to get to really quick. So quickly, is the support guide free to download? I assume it is. Yes.
Free download. Absolutely.
Okay. All right. And one of the questions that came in is that this test, and I do believe this as well, can be pretty overwhelming, not only for providers, but also patients.
Yeah.
So, and one of the participants basically said, people, especially patients, spend time chasing the green.
Yeah.
What do you say to that? Are there patterns? Are there way to start? Are there high levels where you're like, look, we have all this data. Let's do this 1 and then 2 and then 3.
Yeah.
Give me that.
No, that's great. Because having used this with, many, many patients myself, having a practice and whatnot, I completely understand the question. So first, from the provider standpoint, from learning the test, I'm completely sympathetic there too, because, I started here, and the first time they were like, you're going to have to speak to this on the phones and you're going to get through it in a half hour. It's like, really? And so like, what I would say as far as wanting to learn more or wanting to be fluid or trying to not have it be overwhelming, enjoy your own curiosity with it, right? If you want to get into the weeds of every biomarker, then by all means do it, but you don't necessarily have to, because the roll-up of those first couple pages is really what we're trying to do to make it easier for the physicians and for the patients too. And so look at those main categories of key dysfunctions, look at the nutritional components. And as it relates to the patients, we have a lot of doctors actually that will try to get them to just focus on those first 5 or 6 pages and not the actual data on the back end, and really just tell the patient, look, here's all the kind of like recommendations and what we saw. All the back part, all those marker, all of that is just the data that we used to generate those recommendations. And you don't have to get lost there, right?
Okay.
And then the last thing I would say is like, there's always leaders and followers, and there's headlines, and then there's not headlines. And so like, start with the things that are glaring. And if you're chasing the green, like chase the green on the major things like oxidative stress. We know that's terrible, right? Or whatever shows up on that front page in the red, start there. And then likely things downstream will follow on, right? That's the patient improvement.
Right. Great. Okay. Yeah. So we've got those big things that probably can correct downstream a lot of the other things that are kind of kicking off from a biomarker perspective.
Yeah.
Thank you so much. Okay. I'm going to add there's a couple questions, and it really is between the metabolomics and the NutriEval. Obviously, one is probably it's a send home test with a blood spot, correct? And
Yes.
urine. And then the NutriEval, a little bit more robust where phlebotomy is involved. What's the difference between the two from a data perspective? And then as well, when would you use one versus the other?
Yeah, no, it's great. So a little history around that too. The metabolomics we actually released during COVID times.
Yeah.
Because we knew phlebotomy was going to be incredibly challenging. And so we wanted to be able to have as much as information as physicians could get, but have it be an at-home collection. And so when it comes to the difference, so if you think about the difference between the blood draw for fatty acids and a finger stick for fatty acids, because that's one differentiator, we saw actually incredible correlation between, the blood that's coming from the fingertip and the blood that was coming from the vein. We did many, many tests, side-by-side comparisons, and like the correlation curves were super tight. And that's lab speak for like, it looked really good. And so there's not a lot of difference between that. I'll say that there is a difference in when what we're doing with the blood draw is we're actually looking at the red blood cell compared to whole blood, which comes out of your finger. Kind of a minor nuance that clinically, I don't think it would make a decision point for me. But there were things that we can't get on a blood spot. Vitamin D, glutathione, CoQ10. Naturally, those are antioxidants that they break down. They're very labile. They're hard to keep a hold of, and they don't like sitting around and, on a card for a long period of time. So there are certain things that we can't get on the metabolomics. So if you want the whole kit and caboodle, if you want glutathione, CoQ10, vitamin D, you're probably going to have to go with the NutriVal, or you are going to have to go with the NutriVal. If you're willing to sacrifice a couple of those things for the convenience, metabolomics is great too.
Okay. Excellent answer to a question I think is super important.
Yeah.
This comes up for my practice quite a bit when I use this test, but for you, do patients need to discontinue supplements before the test?
So we get this with most of our testing. It's always interesting because there's been a lot of pivot around what we say too, just trying to think about it logically. But if you have one of those patients that comes in and they've been on this and off this, and they've been experimenting themselves, and they've got the grocery bag of different supplements and what have you, we know who we're talking about, right? And like, they just need to like wash out so we can see what the system looks like without all the tinkering. And so my recommendation is for those people, I typically will say, hey, let's get a baseline. Let's have you stop everything. And just remember, there's vitamins that are water soluble and ones that are fat soluble, and they wash out at different time periods. So, B vitamins, you could probably stop for 4 days, and then everything's going to be kind of considered to be washed out of the system. But if you're talking about omega-3s, that's going to take a little bit longer. So you have to be a little choosy, a little picky around what you recommend. Me personally, like I said, if you've got that complex case with all the different tinkering, I will normally recommend discontinuation for a week before running a baseline, still knowing if they're taking something like a fish oil, it might show up on the test. But then for a follow-up, then I want to see if it's working. So I don't have them stop for a follow-up.
Okay, good. And then I'm gonna coattail on that comment. So what do you suggest as far as follow-up? And I mean, obviously this is cost prohibitive perhaps for some people, as far as like a follow-up for a redo of the test. Once you have seen the data, implemented some of the strategies, what do you want to see next?
Yeah, it's great. And I normally base that a little bit on clinical severity, right? If I'm working with somebody who really needs a lot of help, then I might think of a little bit sooner of a retest, like a 3-month window. But if I'm just getting started with somebody, got their baseline, I might test again in 6 months to say, hey, am I seeing some improvement here with the therapies I've done? A lot of people will use this as a yearly test as well. You made a great point about cost prohibitive. And one thing to note too, for follow-up, we do have individual sections that you can run if you don't want to run the whole thing, right? So if somebody's organic acids were really problematic, or it looked like their B vitamins were all messed up, we can just do organic acids. It's called the organics. So you can follow up with more of a tailored part of the test where you found the most problems, if that's going to be, more helpful for the patient's pocket.
Oh my goodness. Okay. I am so sorry, everybody, because we have so many other questions and I can't get to them because of time, but thank you so much for them. But guess what, Dr. Chapman, you've been amazing. Thank you all for attending. If you didn't get those questions answered, I just want you to know that you can contact Genova either through their email or contact number listed on the slide. And we are going to take a 5-minute break before you come back, see me again. And then I really do encourage you, this next session, Breaking the Cycle of Mitochondrial Dysfunction, that's going to be presented by Dr. Sarah Douglas, and is a fantastic follow-up to what we've covered so far. So don't forget to exit this session, and then you got to go back to the main menu to get back into the next section. And again, Dr. Chapman, such a pleasure to be with you. Thank you all very much.